Hi, my name is Beate Kern. I'm a molecular biologist by training. I studied here in Munich, where I also did my PhD research in microbiology, actually working on Helicobacter pylori on the cell biology side. So a bit exotic. I've been with NanoTemper Technologies now for 2.5 years. I started in customer support, and now I'm part of the product management team. NanoTemper Technologies has been around since 2008. Our goal is to enable researchers to do the best science of their lives. So we're really passionate about science and research, and we want to help people really be more efficient and do good research easy and fast. We have two major product lines out there. One of them is called monolith. So monolith instruments are for looking at molecular interactions, so binding affinity measurements. And the other product line is the Prometheus line, that looks at protein stability, more exactly thermal stability, and which, well, you can look at it from a conformational standpoint and a colloidal standpoint. So we look at unfolding and we look at aggregation of the protein while heating it. That can be really important to look at it from different sides, and it enables really a detailed characterization of your protein stability. What I want to show today is this poster that talks about different examples of Prometheus applications in the workflow of drug discovery and development. Of course, this process, the drug discovery and development process, is really long and really complicated. It has many steps, and the more you can improve on it, the better. It always helps those researchers at that step to do better research. And we found that we can actually use the Prometheus in several places there. They're all lined up on the poster, as you can see. And I want to highlight a couple of those today. So the first one is a project that we did in collaboration with Boehringer Ingelheim, where we looked at long-term stability of biologics. You know that biologics, or any type of protein, of course, can be tricky because they're not necessarily stable over time, but you need them to be stable. If it's a drug, you need it to be stable for a while. So you have to find those that can be stable, and you have to find the right conditions to keep them stable, the right buffer, the right formulation. What we did with Prometheus was we performed some experiments all within a day that are potentially able to substitute the standard workflow at Boehringer Ingelheim that takes them 17 months. So that would be great, of course, and would enable people to really only use with the work with the promising candidates and not waste time on those that don't make it in the end. So what they commonly do is you take a protein, you look at it now, and then you wait for 17 months, and then you look at it again and see if it's still okay. Of course, that takes 17 months. The Prometheus was able to look at the same thing in a day, looking at thermal unfolding of the protein, at thermal aggregation, and at chemical unfolding also. So by using these three different points of view on protein stability, we were able to look at different formulations for a protein and rank them, which one keeps the protein most stable. And we got the same ranking that Boehringer also got with their standard workflow of looking at protein stability after 17 months. So this can really help predict long-term stability and can really help save a lot of time, effort, and money, of course, in this research workflow. The second example is a comparison. So we want to compare Prometheus to other methods out there that do similar things. This is about microDSC. That's also a technology for looking at protein thermal stability. And you can see that we compare, of course, really well. The Prometheus gets the same kind of data and gets the same quality of data as DSC, but we get it using a lot less sample and a lot faster. So that's really great benefits of the Prometheus. And we hope that more people can benefit from that to make research more efficient. And the third example is one that is not just relevant to people working in drug discovery and development, but to pretty much anybody working with proteins, because it's a general look at keeping proteins happy. So you can use the Prometheus to look at different buffers, for example, different storage conditions for your protein, different stress treatments, and so on, that you can really look at the protein, use the best conditions for it, and make sure it's happy for the experiments you want to do after. So summing all of that up, of course, I hope that I could show you that the Prometheus is useful in many, many places, not just, but also in the drug discovery and development workflow. And if you feel free to have a closer look at the poster if you've gotten curious, or if you have any other question, feel free to contact us. Our website is nanotempertech.com. and we're always happy to help. We're all scientists ourselves, and we know what it's like to be in your shoes, and we'll try our best to find good solutions for you and your research. So I'd like to thank you, and I'm hoping that we'll be in touch. Bye-bye.